All right, so I'm excited for today's call because we are covering brand new research today, like literally hot off the presses, on a topic that has been semi-controversial in the past, but is also very interesting to two groups of people. People who have a history of Epstein-Barr virus, especially people with chronic active Epstein-Barr virus, and people with multiple sclerosis. And then I guess the third group would be scientists and doctors and people like me who, are trying to figure out connections to all this, and especially causal connections. And so with multiple sclerosis is a neuroautoimmune disease that affects the central nervous system. And there's no known cause in the research. If you read about it, they'll say it's idiopathic, which means they don't know the cause. And this new research is suggesting that Epstein-Barr virus might be a causal factor and is the strongest study to date supporting a causal factor for Epstein-Barr. So we'll jump into that today. But first, I want to discuss Epstein-Barr virus and multiple sclerosis separately before we combine them and look at this new study. So let's jump over to the whiteboard. So multiple sclerosis, like we said, is a chronic neuroinflammatory autoimmune disease. And what it does is it targets the neurons of the central nervous system, specifically myelinated neurons. It's known as what's called a demyelinating disease. So what does that mean? If we draw a neuron here, so we'll go like this. Here's the branched end. Okay, so this is a nerve or a neuron, and your spinal cord and your brain are made up of individual neurons. So I'm sorry if this is elementary, but I just want everyone to be on the same page. And so let me clean this up a little bit. Oh, that's not what I wanted to do. Undo. Love that feature. All right, so here's our neuron. Now, this is an unmyelinated neuron. This is what would be called a C fiber, which C fibers transmit pain and tickle and achiness and things like that. C fibers compared to A fibers, which are myelinated, are very slow. So unmyelinated C fibers are very slow compared to myelinated A fibers. A fibers are fast because they are myelinated. And the difference is A fibers or myelinated neurons are going to have things called myelin sheaths on them. So these blue squares represent the myelin sheaths. And so the difference between the two in terms of transmission of a signal is if we undo those myelin sheaths and we go back to a C fiber is going to transmit a signal like this, right up the center, slowly to the other end. And then at the dendrites here, it's going to connect to other nerves that are branching off and transmit that signal to those other nerves. Okay, so it's slow. It has to travel the whole length of the axon, which is that long, narrow tube of the nerve. If we then go back and we go to a myelinated nerve, the myelin helps speed conduction of the nerves because the signal now doesn't have to go long and slow along the axon. It can jump, boom, boom, boom. So it goes fast. It gets there quickly because the myelin creates what are called these nodes of Renoir, which the signal can just jump node to node. It gets to skip, right? So rather than having to travel along the entire highway, we can kind of skip, hop, and jump along. And so those myelin sheaths promote speedy conduction. And A fibers are what transmit, pressure and certain sensory inputs and help us, think and respond and reflexively react quickly. So if we start to have an autoimmune process that attacks the myelin sheath, a demyelinating disease, now we lose the myelin, and where this used to do that, now it has to go slow, and it has to travel slowly along a nerve that it's not supposed to travel slowly on. So now you may lose symptoms, or you may have symptoms of MS, like poor balance, or speech issues, or recall issues, or vision issues, or it just depends on what part of the brain or spinal cord are being impacted by the disease in that given individual with MS. So this is excuse me if this picture looks sloppy, but this is we'll call it a kindergarten drawing of the disease process for you. I just wanted you to understand the neurology that's affected there. Now, Epstein-Barr virus, if we go over here, what color should we use? Let's use brown for Epstein-Barr virus. So Epstein-Barr virus, we'll call it EBV for short. Epstein-Barr virus is the virus that causes mono. Okay, so if you've ever heard of someone who has mono, or you've had mono yourself, mono is short for mononucleosis, and mono is the extreme end stage result of Epstein-Barr virus infection. So, you know, if you've had it, you may have had, you know, the typical or the stereotypical mono case where you're laid up in bed for a month. You know, you contracted in high school or in college, and you're laid up for a month, extremely fatigued, missed your classes, literally couldn't get out of bed. That's mono. Well, mono is caused by Epstein-Barr virus. It's possible to have Epstein-Barr viral infection and never have mono, right? The similar explanation would be it's possible to have blood sugar issues and not have diabetes. Understand? There's a whole spectrum of potential symptoms and outcomes. So Epstein-Barr virus is a herpes family virus, and herpes family viruses, once you are infected with them, they live in the nervous system forever from there. And Epstein-Barr virus specifically likes to live in B cells or B lymphocytes in our immune system. And so what happens is, if we tested the planet, 95% of people tested would show a history of Epstein-Barr virus. Okay, so almost all of us have been exposed. And if you get exposed as a young child, you never have symptoms. It's not unless you aren't exposed until adolescence or later that you may have mono or other symptoms. And it's spread via saliva. So, right, mono is the kissing disease. So, you know, family members can spread it to each other, just kissing each other goodnight or whatever. So 95% of the planet would test positive for history of Epstein-Barr. But so we've all largely been exposed to it. So then it's living in our nervous system and our B lymphocytes, and it's up to the immune system to keep it latent. Okay, if it's latent, it's dormant. All right, it's in there. It's kind of like a hibernating bear, and as long as you don't wake up that bear, you're safe. But if you wake up that bear, now that infection can be what's called reactivated. Okay, reactivated, and a reactivated infection can have consequences. You might reactivate it and have mono again, or you might not hit mono, but you have reactivated Epstein-Barr and you're having symptoms like fever, undulating fever, swollen lymph nodes, maybe fatigue, maybe sore throat, you know, lots of quote, maybe flu type symptoms that you might think are flu or might think are strep throat, but it's actually Epstein-Barr. And you go to your doctor and they test you for flu and you're negative, or they test you for strep and you're negative, and you're frustrated because you're like, but I have a sore throat or I have flu-like symptoms. What's going on here? Well, they may even test you for Epstein-Barr, but there's a lot of in conventional medicine, there's a lot of misinterpretation of Epstein-Barr labs that can cause you to miss it. Okay, and so that goes to a conversation of antibody levels. And so when we talk in conventional immunology, you're taught that we have 4 different antibody classes or immunoglobulin classes. So there's IgM, IgG, IgE, and IgA. Whoa. Okay, and so IgA deals with mucosal immunity, gut, lung, sinus, bladder, anywhere you have mucus. We're not going to talk about that right now. IgE deals with allergy, from hives to anaphylaxis. That's different videos. We're not going to talk about that right now. Relevant to the Epstein-Barr conversation are M and G. So IgM, think Marines. These are the first antibodies produced when you have a new exposure, a new antigen, or a new foreign object. That could be a first-time exposure ever to a virus like Epstein-Barr. That could be a first-time exposure to an environmental allergen like, poison ivy or ragweed or whatever we're talking about. That's a first-time exposure. So you're exposed, IgM within 3 to 5 days ramps up to show acute exposure. And then a few days later, IgG levels peak. And IgG levels, if M is the Marines, are the first ones on the scene, kicking butt and taking names. IgG comes and they have the body bags. They're cleaning up after the IgM. Okay, and then the IgG eventually become our memory cells. And this is important because they remember that we've seen Epstein-Barr before. So if we have Epstein-Barr again, if we have a reactivation, the IgG will clone out right away and start that response. And you almost never have a second IgM response to something you've seen before. It's usually a reactivation or a cloning out of IgG again. So conventional immunology looks at IgG as just memory. Oh, you've seen that before. But more up-to-date immunology says that presence of IgG, yes, indicates memory. You have seen it before, but depending on what your IgG levels are, it could also mean reactivated virus. Okay, and so here's where the issue of interpretation is. In most doctors' offices, you might think you have Epstein-Barr, and they'll run things like Epstein-Barr virus, viral capsid antigen, IgG in your blood, and they'll run the same thing, IgM, and the IgM will be negative, so they'll say, oh, you've just seen it in the past, it doesn't matter right now. And there's another one called Epstein-Barr EBNA, which are antigens, nuclear antigens, and those are IgG. And so typically, if you've had Epstein-Barr, if you're one of the 95% of the planet that have been exposed, both the VCA IgG and the EBNA IgG are going to be lab high, and they're always going to be for the rest of your life. So your doctor will see that and say, okay, well, that doesn't matter. That just means you have memory. But depending on the titer level of those, one or both of those could signify reactivation for us. But there's a lot of gray area there. So what we'd like to see is also you run an Epstein-Barr virus early antigen, or EA IgG. And this one, if this one's positive, this one is telling us reactivation. Okay, reactivated infection, clinically relevant infection. Because if the early antigen is being shown, it means the virus is in the lytic phase and it is actively hijacking your cell's machinery to reproduce and make other virions, viral babies. Okay, so unless your doctor understands how to interpret these things, they may miss that you have a reactivated Epstein-Barr virus infection. And most doctors and most quote unquote Epstein-Barr panels do not include the early antigen marker. So you might think they ran a thorough test and they didn't. So again, you're given the wrong information. You're walking around with active Epstein-Barr virus and don't know it. Okay, that's important to know whether or not you have MS or MS symptoms, because Epstein-Barr virus alone can wreck your life, regardless of whether or not you have a neuroinflammatory autoimmune demyelinating disease. Okay, so this is the background to the Epstein-Barr and the MS discussion. Here we are with the paper, and this is published in the journal Science, and it just came out. Okay, it just came out, I believe it was January 21st, excuse me. And so you can see the title here is Longitudinal Analysis Reveals High Prevalence of Epstein-Barr Virus Associated with Multiple Sclerosis. And that's not as strong of a title as I would like, but I didn't write it. So you can see in the abstract, multiple sclerosis is a chronic inflammatory demyelinating disease of the central nervous system of unknown etiology. Again, they say unknown cause. We tested the hypothesis that MS is caused by Epstein-Barr virus in a cohort comprising more than 10 million young military adults in active duty in the US military. 955 of those 10 million people were diagnosed with MS during their service in the military. So they entered the military without MS, and while they were in the military serving, they were diagnosed with MS. So we have data on them pre-MS diagnosis and then afterward. And what they found was risk of multiple sclerosis increased 32 times. You have a 32-time greater risk of MS development after Epstein-Barr virus compared to any other infection. So they found a 32 increased risk of MS with Epstein-Barr, but no increased risk from any other infection, including other herpes family viruses such as cytomegalovirus. So that's important because herpes viruses are everywhere. Cytomegalovirus is one, herpes simplex 1, herpes simplex virus 1, herpes simplex virus 2, human herpes virus 6, 7, 8. There's lots of them. So none of the other herpes family viruses increase risk of MS, but Epstein-Barr did by 32 times. They also found that the blood levels of neurofilament light chain, which is a marker of axon degeneration, that skinny long part of the neuron that the signal has to travel on, the levels of that in the blood was increased only after Epstein-Barr infection. None of the other infections. So they end with, these findings cannot be explained by any known risk factor for multiple sclerosis and suggest Epstein-Barr virus as the leading cause of multiple sclerosis. Bam, that is huge. They didn't say it's associated with, they didn't say it's correlated with, they said it suggests it's the leading cause of MS. So if we dig into a news article on this from Science Daily, they interview the author of the study and they say more. So the author says, the hypothesis that Epstein-Barr caused MS has been investigated by our group and others for several years, but this is the first study providing compelling evidence of causality. This is a big step because it suggests that most MS cases could be prevented by stopping Epstein-Barr virus infection, and that targeting Epstein-Barr virus could lead to the discovery of a cure of MS. Epstein-Barr virus infects approximately 95 of adults, and MS symptoms begin about 10 years after Epstein-Barr infection. So again, here's another reason why there may be a disconnect between Epstein-Barr infection and MS being causally associated, because clinically there's 10 years between when you had Epstein-Barr and when you had MS. And so it's easy for doctors to not put that together, even though you ever had Epstein-Barr, or for you may have forgotten you had Epstein-Barr, right? So there's lots of ways that connection may not be made. And another one that the author of the study said is that the delay between Epstein-Barr virus infection and the onset of MS may be partially due to the disease's symptoms being undetected during the earliest stages, and partially due to the evolving relationship between Epstein-Barr and the host immune system, which is repeatedly stimulated whenever latent virus reactivates. So basically the delay between Epstein-Barr infection and MS diagnosis is partially due to MS symptoms being undetected in the early stages, right? You might just think you're dizzy, or you might just think you're having some vision issues. It might take years of damage before you finally are like, wow, this isn't, I can't bear this anymore. I think something's wrong. I need to go get it checked out. So this study is very powerful because they had people with no MS and no Epstein-Barr virus infection pre entering the military. Then through their service in the military, they were diagnosed with MS, and the blood work connected the Epstein-Barr virus in the blood with their MS diagnosis. So that's powerful. The thing I don't like here is that they say currently there's no way to effectively prevent or treat Epstein-Barr infection. That's not true. And they say, but an Epstein-Barr vaccine or targeting the virus of Epstein-Barr antivirals could ultimately prevent or cure MS. That's what they always say, right? But there is no Epstein-Barr vaccine, and viral vaccines have been an epic failure throughout the history of vaccine science. And then antiviral drugs, there's lots of antiviral drugs that are used for herpes simplex viruses that are not effective for Epstein-Barr. So the likelihood that they're going to develop one that is low. Also, there's plenty of scientific evidence supporting different herbs that do effectively address Epstein-Barr virus. So let's look at some. And the major one we want to look at is andrographis. So this is a study from the Archives of Virology titled Broad Spectrum Antiviral Properties of Andrographilide, which is found in the herb andrographis. And so if we go down here, I'm going to show you a graphic here. So this is the effects of andrographis on the immune system and virus entry and propagation. And so all these things are showing you benefits of andrographis. So it inhibits herpes simplex 1 protein synthesis. So there's a herpes family viral member that it inhibits. It reduces swine flu. It suppresses hepatitis C virus. It increases CD4 cells in HIV patients. It inhibits human papillomavirus gene expression. It restores tumor suppression gene in human papillomavirus, okay, which is the virus tied to cervical cancer risk. It inhibits hepatitis B virus and herpes simplex 1 DNA replication, inhibits hepatitis C virus protease and Epstein-Barr lytic protein expression, okay, inhibiting Epstein-Barr metabolism, inhibits herpes simplex 1, inhibits herpes simplex 1. So, wow, we just said currently there's no way to effectively prevent or treat it. Well, if we can inhibit the function of the virus, you are preventing it from taking over your body and you are treating it. So that's not true. It's just that effective natural strategies don't get the press because they don't make the money, right? So there's a graphical representation of how andrographis works. And here's a different picture. So this is showing how it works to inhibit the viral life cycle. And there's andrographis can inhibit the viral entry into the cell. It inhibits its protein synthesis, so it can't be replicated and sent out. Okay. And then if we look at studies specific to Epstein-Barr virus here, let's change how we're seeing this page. So here you see Epstein-Barr virus, and we're seeing the antiviral effects of andrographis on different viruses. So for Epstein-Barr virus inhibits 2 viral immediate early genes, resulting in inhibition of viral lytic protein expression. So what does that mean? That's basically saying, if we switch back to the whiteboard, that andrographis is inhibiting early gene expression, bang. Which, remember, when early antigens are positive, that's reactivation and suggests that the lytic phase of the viral cycle is happening. So that study shows us that andrographis is actually an effective preventative and treatment strategy for Epstein-Barr virus. So if we could leverage andrographis to inhibit this, then we inhibit the driving of MS. So if this paper says there's a causal link between Epstein-Barr and MS, and we can inhibit Epstein-Barr and prevent it, then why doesn't that result in prevention or inhibition of MS development? Powerful stuff. Other herbs that are shown to be antiviral and have effectiveness against Epstein-Barr virus. Another big one is astragalus. So remember the 2 A's, astragalus and andrographis, are very powerful antivirals in general, and andrographis specifically for Epstein-Barr virus. So if you're out there with MS and you have a history of Epstein-Barr virus, obviously it's too late to prevent MS, but if you have a history of Epstein-Barr virus, Epstein-Barr virus may still play a role in your flares, in your risk of flares. So promoting an immune system that keeps the Epstein-Barr virus latent is probably a very high priority clinical goal for you to prevent MS flares and MS progression. So this is one potential, this is one strongly supported causal factor of MS. Are all MS cases caused by Epstein-Barr virus? No. But again, if we're working with a client with MS and they have a history of Epstein-Barr, that will be an area of strong clinical focus likely. If you're someone with a family member with MS and you don't have MS and you fear developing MS, then understanding your Epstein-Barr virus titers and having optimal antiviral immune defenses is going to be a key player in helping prevent you from developing MS. One other nugget to add to this is MS isn't the only autoimmune disease that Epstein-Barr is implicated in. It's also strongly connected with Hashimoto's disease or thyroid autoimmunity, and others like Sjogren's disease. There's multiple that it's connected with. So, Epstein-Barr is not something we want to let run rampant in our bodies.